in this video, we will study about structure of pollen grains. Pollen grains are produced in the flowers from the anther part of androecium. This is the anther part, like we have discussed in the previous videos. If we see the anatomy of anther, now this part is called as sporogenous tissue. This is surrounded by a covering called as tapetum. Now there are in diathecous anther there are four group of sporogenous tissue. Now this sporogenous tissue, this get differentiated to form spore mother cell. This tissue is diploid, spore mother cell are also diploid, and when this undergo meiosis, right, this will produce haploid microspores. Now microspores will be produced from the sporogenous tissue and they will be haploid. Now these microspores, they are also called as pollen grains. Now they are being produced after meiosis, so their genetic constitution is haploid in nature. So pollen grains, they are haploid, and color of the pollen grains is yellow. This yellow color is due to the presence of flavonoids and carotenoids. Their mixture make the color yellow in color. Now, cytoplasm of the pollen grain is rich in reserve food material, starch, fats, and unsaturated oil. Now, this production of pollen grains or microspores from sporogenous tissue, this is also called as Microsporogenesis. Now, these pollen grains, they are of different shapes. They have we can see here in this figure, they are of different shape. They may be globose, they may be spherical, they may be oblong, they may be oblate, and their outer covering, this is the outer covering of the pollen grain. We can see this has different structure and architectural pattern. This may be formed of ridges, tubercles, spines, reticulation network. So different shapes are there, right? We can see here tubercles are there, then in certain cases reticulation may be there, right? So this is about different shapes as well as their pattern of exine, which is the outermost covering. This shows different patterns. Now this exine may be made up of sporopollenin. Now what is sporopollenin? This is oxidative polymer of carotenoids plus carotenoid esters. Exine may be rich in proteins. Now, when this pollen grain fall on the stigma, reaction will take place between the pollen grain and the stigma, right? So this is called as compatibility reaction. So these chemicals help in identifying stigmatic surface and pollen grain and helps in their germination. So exine may be rich in proteins. Exine is basically derived from tapetum. Now this may be made up of Yubish bodies. Now what are Yubish bodies? These are acellular structure. Again, they are part of the sporopollenin, rich in lipids, and they also help in formation of the exine. Now we can see here I have made this in this diagram. This part is tapetum, right? And this part is sporogenous tissue. Now when tapetum, now this is tapetum, and when spore mother cell develops into microspores or pollen grains, now this tapetum will help in formation of the exine covering. Now this may become like this and they get fused to form a structure, or this may be secretory in nature. We have discussed in the previous videos. So this will get deposited, this tapetum secretions, or this may get deposited forming a plasmodium like structure around the microspore or the pollen grain. Then this helps in formation of the exine. Now this tapetum is diploid in nature. So this outer covering exine will also become diploid in nature, because this outer covering of the pollen grain, this is being produced from the tapetum tissue. Tapetum is the outermost layer of the sporogenous tissue, which helps in its nourishment and also helps in deposition of the proteins, sporopollenin, Yubish bodies. So this will lead to the different architectural design of the exine of the pollen grains. So this is about different kinds of pollen grains. They have different shape and they have also different sizes. They may be small in size, they may be big in size. We can see here these are different kind of pollen grains, and here these are shown thin areas present in the pollen grains, and these thin areas are also called as germ pores. Now discuss about structure of the pollen grain which is being produced after meiosis. Now this is covered by outermost covering, and this outermost covering is called as exine. Now this is the outermost covering, exine, but at certain places this may be thin, and this is called as germ pore area. Like we can see here also, these are germ pores. Now these germ pores may be present in the form of a pore, or they may be in the presence of a furrow-like structure. When it is pore, this is called as germ pore. When it is in the form of a furrow, this is called as germinal furrow. Now this area, this internal wall is entine, this is thin, and this is made up of pectin plus cellulose. So this is called as pectocellulosic in nature. Now this is the cytoplasm having tiny vacuoles, and this is the nucleus of the nucleus present in the cytoplasm called as tube nucleus. This shows convolutions at maturity, and this is the generative cell which has become free from the internal wall of the pollen grain. So at this two cell stage, now these anthers may help in liberation of the pollen grains. Now anthers, now this will days open to liberate the pollen grains. So at this stage, at the two cell stage, that is when it has tube cell or generative cell, this may be liberated from the anther. So this process is also called as pollination. Now if we see the fine structure of the spore wall of the pollen grain, now this is the outermost layer called as exine. Basically this is being produced from the tapetum region present around the sporogenous tissue. Now this fine structure, if we see, this is made up of this covering. Up to this covering is exine, and this is the entine. Entine is pectocellulosic in nature, mean this is made up of pectin and cellulose. Now this part is exine, and exine is made up of exine called as sexine or exine. This is exine, and this is exine or sexine. Now we can see here exine, entine. Exine is outer exine, inner exine. This is also called as sexine, nexine. Now this outer exine, this region, this is made up of a foot layer. This is foot layer. These are rod-shaped baculum, and this is called as tectum. This may not be continuous. Here it is shown continuous, but this may not be continuous. So this is made up of outer tectum, baculum, and inner foot layer. So this is exine, this is exine, this is the entine part, and this is also called as sporoderm of the pollen grain, that is exine or outer covering of the pollen grain. In pollen grains which are pollinated with the help of insects. So when they are pollinated with the help of insect, this is called as entomophily. And such pollen grain which are carried by insects are called as entomophilous pollen grains. Now these pollen grain which are specially pollinated with the help of insects, they have pollen kit, and their pollen kit is made up of lipids plus carotenoids. Now pollen grain have specific number of thin areas. Now these thin areas may be in the presence of pores, or they may be present in the form of furrows. When pore called as germ pores, and when present in the form of furrows called as germinal furrows. Number may be 1 in one pollen grain, then it is called as monocolpate, and number may be 3, then it is called as tricolpate. Like here we can see in this figure, this pollen grain is tricolpate in nature. These are the thin areas present in the pollen grains. Now, when we study the pollen grains of various aspects like its exine covering, germ pores, internal study, so this study is called as Paleonology. Now, this Paleonology is also of different type. When we study the pollen grains in fossils, various kind of fossils, like long time back, when plants were present and they become extinct and their pollen grains have been deposited inside the rocks or certain sediments, right, they are called as fossils. So this study of pollen grain in fossils is called as geopaleonology. Now when pollen grains, they are dispersed with the help of air from the anther, and in the air certain pollen grains are there, and their study in the air is called as aeropaleonology. Supposing certain criminology has occurred or crime has occurred, so pollen grain may stick to the body of the animals or human beings or criminal. So that study in the medical aspects of criminology in pollen grain, this is called as latropaleonology. And honeybees, they sit on the flowers and then they eat the nectar, and then on their legs, pollen grains are attached. So when they form honey, inside pollen grain may be there. So this study of pollen grains in honey is called as Paleonology. So this is different type of paleonology. Uh, Now these pollen grains, they are the first cell for the gametophytic generation. So they help in the formation of male gametophyte. That's why anthers, they are called as male reproductive organ of the plant. And this formation of male gametophyte from the microspore or pollen grain, which we will study in the next video, this is also called as microgametogenesis. Like first we discussed microsporogenesis, that is the production of microspores from the spore mother cell by meiosis. Now this is the production of the male gametophyte from the microspore or the pollen grain, and this will happen with the help of mitotic cell division. This is called as microgametogenesis. So these are the first cell for the formation of the male gamete, or first cell of the gametophytic generation, that we will study in the next video. Thank you for watching my video. If you like my video, please like, share and subscribe. If you have any questions or doubts, please ask me in the comment box.